Hello everybody, my name's Joe and I'm a naturopathic student based in Italy. The date today is the 5th of May 2020. So like everyone, I've watched this coronavirus situation unfold here and watching as it continues to develop across the world. But I've resisted making a video so far because there is just so much information out there and it's all changing so quickly. But I've decided now is a good moment to put this out because I believe we're missing a large part of the conversation from the mainstream. And that is, what can we do in our daily lives to build resilience and reduce risk, not only from getting sick from the virus, but from ill health and disease in general. Because what we're seeing is that COVID-19 is part of something much bigger. It's happening on a backdrop of ill health and chronic disease. A recent study out of Italy looked at over 16,000 COVID-19 deaths and found that only 3.5% of subjects didn't have a pre-existing chronic disease. Now, of course, this isn't everyone, and there's still a lot we don't understand. This is not about trivializing things, quite the opposite really. If it turns out that chronic disease is causative to this degree, then dealing with it will be an essential part of the solution. And even if, at the end of all this, the chronic disease link turns out to be only correlation, putting our focus on improving chronic disease has the capacity to completely wipe out the economic damage done by COVID-19, as well as free up our healthcare systems and of course save millions of lives. Pretty good side effects. Chronic disease kills millions every year, puts a massive strain on families and healthcare systems, as well as our economy. COVID-19 is set to cost us an estimated $1 trillion globally, and of course that is massive, but the US alone spends more than that every year just on dealing with chronic disease. One report estimated that the global cost of chronic disease between 2010 and 2030 is going to be $47 trillion. So at worst, if we take action here, we will save lives and the economy. At best, if we really focused on all those things which drive chronic disease, not only directly on diet, exercise and sunshine exposure, but also economic disparity, pollution levels and environmental issues and so on, well, we'd be looking at a totally different world. But isn't chronic disease inevitable? The real issue here is that when we think about these pre-existing diseases that we've seen in COVID-19, hypertension, cardiovascular disease, diabetes, obesity and so on, they are largely preventable. Of course, genetics and increasing lifespan are important, but they don't explain the recent surge in chronic conditions in young people all over the world. What is surely more important is how we live, what we eat, our relationships with each other and with the natural world. What we see is that these modern diseases are driven by our modern lifestyle. Our food supply, air, water, electromagnetic spectrum, our way of life, all these things have been industrialized, especially in the cities. Air pollution alone kills 5 million a year globally. On one hand, industrialization has brought many benefits. It's lifted billions out of poverty, it's improved the standard of living in incredible ways, but it has a cost. It is also driving metabolic dysfunction, low-grade inflammation, oxidative stress, insulin resistance, leptin resistance, and many other underlying mechanisms behind chronic ill health. So while this situation is tough, it's also bringing awareness to all this. It's given us a glimpse of more peaceful cities with less air pollution, grounded our planes, shown us the importance of connectedness and community, of how precious our healthcare workers are. It's questioned our economy, this throwaway culture of endless growth and waste. It's put these chronic diseases at the fore and, at the heart of it I guess, it's questioned our fundamental relationship with the natural world. So in my opinion, there's never been a better time to start addressing some of these underlying drivers. We need to stop looking at everything in isolation and look at the bigger picture, to stop going after symptoms and start dealing with the causes. So going forward, I'll be talking about the effect of these lifestyle factors: sleep, sunshine, diet, physical exercise, stress and toxic exposure, not only on helping our immune system do its job, but also on the prevention of chronic disease. So before we get into it, it's important to say be critical. Be critical of what I have to say, of what you hear on the news and in conversations and social media and so on. And I don't mean be dismissive, I mean let's try to be objective about what we hear. Everyone has their bias and their beliefs, and in times of stress the temptation is to seek out information that confirms these biases instead of challenging them. But I think if we are aware of this, in ourselves and in others, then we can learn from each other and move forwards together. So to be clear, I'm not a doctor, I'm not an immunologist or an epidemiologist. I don't have any formal medical training, so please keep that in mind. We are all learning here, and my main motivation is really to highlight these things rather than to give any specific recommendations. For that, please get the help of a qualified medical professional. And of course, if you feel ill or suspect you have the virus, then seek medical help, not only for yourself, but also to protect those around you. I'm not going to be talking about treatment options for this virus, but instead on looking at prevention through lifestyle changes. So I'll start with some general information about the immune system and some characteristics of this virus, to give context to the lifestyle factors that I'll be talking about later on. So why do I want to focus on the immune system? Well, we live in a microbe-rich world. We've evolved in a microbe-rich world, and as such, our immune system has evolved all sorts of different tools to deal with these microbes. And we see that in the current situation as well. The majority of people who get diagnosed with COVID-19 go on to get better on their own without the need for hospitalization and without particularly severe symptoms. Now, that's obviously not to trivialize the people that go on to need hospitalization and go on to need ICU care, and some who unfortunately go on to die from this thing. And of course, for them, it's a very, very serious situation. But I want to say that when the immune system is working well, and when we are fit and healthy, for the most part, this is not giving us too many problems. And so every day of our lives, the immune system manages these microbes. It doesn't just indiscriminately kill everything. It's evolved to manage them in some cases as a resource. So we have our bacteria, fungi, viruses on us and in us that act as symbiotic organisms, and the immune system keeps them in check. That's its main job. Of course, if we come across pathogens, then it can neutralize these. It can neutralize also cancerous cells, which it does every single day of our lives. And it also removes toxic and metabolic wastes as well as old cells. And so if we could just pull out the immune system from somebody, they'd probably start to feel pretty bad within a few hours and maybe die within a few days. And so this is one reason why I want to focus on the immune system, because perhaps we don't appreciate all of the work that it does every single day of our lives to keep us healthy and happy and alive. And the second reason is that perhaps we don't appreciate the effect of our day-to-day behaviors can have on the way that our immune system can do its job. So about 8% of our DNA is viral in origin, and that's been linked to both positive and negative health outcomes. But just to say that we've been evolving alongside these organisms for a long, long time. More than half of our cells are microbial, i.e. not us. And of course, when we look at our genome, our own genome dwarfs in size compared to our bacterial genome, our viral genome, our fungal genome, our mitochondrial genome. And so we are a community. And just recently, viruses have also been included in this symbiotic community, according to researchers. And even our brain may have this microbiome. So until recently, we thought that our brain was sterile, but now we understand that there may be bugs that live symbiotically also there. So our microbiome is an essential part of us. We are a host for the bugs, and the bugs do various jobs for us. They extract calories from our food, they create vitamins, they are good for our metabolism, and they also help us, they also protect us from pathogens. And so maintaining that symbiotic relationship is a really important part of our physiology. So how does the immune system deal with problematic viruses? Well, the first thing it can do is to create a fever, and that rising body temperature can disactivate viral enzymes and viral replication. And so that's obviously really important. But it also stimulates the adaptive and the innate immune responses. So our innate immune response is our early, non-specific response. And when we're talking about viruses, this consists mainly of interferons. And so what happens is that our immune cells sense danger patterns or microbial patterns, and they do that using various receptors called PRRs, and that triggers the release of interferons and other chemicals in response. So these chemicals are broadly called cytokines or chemokines, and they're basically little signaling molecules that tell different parts of the body to respond in different ways, particularly to threats like infections. And so interferon signaling has the job of decreasing replication, and it does that by obviously, as we said, stimulating other parts of the immune system as well as stimulating protein creation. And so this has the effect of decreasing replication and also changing the expression of MHC. So MHC is a protein complex that we find on the outside of cells, and it basically tells us whether that cell is friend or foe. It tells us whether that cell is healthy and we don't need to do anything, or whether it's infected and we need to call in the immune system to destroy that cell. So then we have our defensins. These are antimicrobial peptides. They're important for the direct response against some pathogens, and they also stimulate various adaptive responses. And then we have our natural killer cells. These are a really important part of our antiviral defenses, as well as our anti-cancer defenses. And once they're activated, they can attack infected cells directly. They produce interferon gamma and other substances to be able to do this. Then we have our dendritic cells. These are kind of signaling cells, and they kind of sit between the innate and the adaptive immune system, and their job is to call in the adaptive response. So then we have the adaptive immune system, and here we have our T cell response and our B cell response. So our T cell response consists of our cytotoxic T cells. These cells can attack infected cells directly when they read that MHC class 1 protein on the outside of cells. And then we have our T helper cell, and with most viruses we see a Th1 response, but it's worth mentioning also that in this particular virus they have noticed some Th2 cytokines also raised. And the job of this Th1 response is to call in other parts of the immune system. And then we have our antibody response. So our antibodies get produced in response to a virus. They are like little protein keys that fit into the locks of the virus, so the virus can no longer gain access into the cells. And obviously viruses need to gain access to the cells because they need our cellular machinery to replicate. They can't replicate on their own. So if we can block them from getting into the cells, this is obviously great at reducing the virulence of the virus. And so this is where we're talking about things like natural immunity. We create a memory of the pathogens that we have fought in the past. And so these antibodies can be produced at short notice so they can disactivate viruses that we see again. This response also has the job of inducing phagocytosis and also activates the complement system. So if we've got all these incredible tools in our immune system, why is it that viruses can still get a foot in the door? Well, viruses are evolving just like we are, and they evolve ways to modulate our immune system. So they're able to turn bits off, and so they can go undetected and go under the radar. And one of the main ways they do this is to inactivate our natural killer cells. So there's a series of proteins called the NKG2 proteins that either activate or inhibit our NK cells, and some viruses have learnt to interfere with these proteins so they can actually go on infecting us under the radar and turn off that NK response, or at least turn it down. And for example, in HCV, NKG2A has been noted as a critical checkpoint in chronic HCV infection. So the HCV virus has evolved to be able to interfere with this protein, so the natural killer cells are inhibited, and it can carry on with a kind of chronic or persistent infection. So I mention this because this was noted in COVID-19 with the SARS-CoV-2 virus. This study looked at natural killer cell exhaustion potentially linked to this NKG2A protein. So in doing that, it suppresses the natural killer cell interferon gamma production. It may also interfere with interferons by changing the STAT-JAK pathway, and this may also lead to adaptive response suppression or delay. And there may also be changes in the TLRs. So these are these toll-like receptors. They're the receptors whose job it is to sense whether there is a threat, either a danger signal or a microbial signal, and then to activate the innate immune system in response to that threat. So if the virus is able to change the way that these TLRs are working, then that's another way that it could get under the radar. And so what about this particular virus? How does our immune system respond? Well, there was a review of a patient by the Doherty Institute, and this patient, I think she was from Wuhan initially, and she was followed in hospital. She didn't need treatment, but she was hospitalized, and so her response could be followed. And what they saw was a fever and a strong adaptive response after 7 to 9 days, which reduced at day 20. And looking through the study, it seems like the natural killer cell activity was blunted. So this may back up what I was saying on the previous slide. Then an article in Nature was looking at functional exhaustion of NK cells and cytotoxic T cells via this increased expression of this NKG2A. So that may be an issue here. Then we are seeing high levels of interleukin-6. This has been a marker that's been very predictive of disease progression. Interleukin-6 is a pro-inflammatory cytokine. It's a marker of inflammation. And so what these researchers saw was that people with higher levels of interleukin-6 were more likely to go on and reach a more severe form of the disease. So this may be mediated by possible changes in macrophages in the lungs. This still has to be kind of nailed down as to exactly what's going on here, but it may be that in the absence of a proper innate response, in the absence of proper natural killer cell activity, macrophages come in, and there are changes in these macrophages, particularly in the lungs, which then produced high levels of interleukin-6, lots of other cytokines, and then we start to see perhaps the tissue damage that is associated with the lung injury and the ARDS that has been coming out. So let's talk about the statistics. So obviously we need good numbers to be able to make good decisions. But at the same time, we know that these numbers are not going to be accurate just because of the difficulty of developing testing and scaling it up enough so that you can test enough people to get a broad idea of what's going on. So obviously the testing has been limited to people who have been presenting with the worst symptoms, and obviously that's going to skew the results somewhat. And so with statistics, we're always looking for the kind of best case scenarios and the worst case scenarios, and using some sort of modeling to find out where we might lie on there. So there are many different factors going into why they may make the situation look better or worse than it really is. And if you're interested in looking into this, I highly recommend a podcast on YouTube by Dr. Peter Attia. He talks a lot about these issues, he weighs up a lot of the evidence, and he gives a really good balanced view, not only about this, but about the situation in general. So mortality rates change based on how we define it, on the number of confirmed cases, as well as political issues, and of course access to medical care. Also, the number of cases depends on the diffusion and the reliability of testing. If we tested the whole country, or if we tested perhaps 20 times more people than have been tested, then we would see very different numbers, and we may see a 20 times drop in the mortality rate. And this may well change the way in which we manage this situation. So without going into too much detail, it's probably safe to say that we're seeing a higher fatality rate, and it is more infectious than influenza. If we look at what happened on the Diamond Princess, where they were able to test the whole population, they saw a mortality rate of around about 1.7, 1.8%. And so this is quite a few times higher than influenza, but it's also quite a few times lower than some of the numbers that are coming out from other countries. And so that number seems to be backed up by a few other studies, and biology seems to confirm this. So what about the biology of the virus? Well, it's important to know a little bit about how the virus works, not only for treatment, but also for prevention of the virus. So one way we can do this is to look at similar viruses to see if there's any common ground. And there seems to be quite a lot of common ground between the original SARS-CoV-1 virus of the pandemic of the early 2000s and this SARS-CoV-2 virus. In fact, that's why it's got the name. And they both seem to gain access to the cells via the ACE2 receptor. The ACE2 receptor is an important part of our RAS system, and that's an important regulator of our blood pressure. And so we find these ACE2 receptors not only on the type 2 pneumocytes. The type 2 pneumocytes are lung cells which produce surfactant, and so they're essential to our ability to breathe. But we also find this ACE2 receptor expressed on lots of different tissues throughout the body. So kidneys, the heart, the digestive tract, other lung cells, even the tongue. And so this may explain the wide variety of different symptoms that people turn up with. So another thing that it may have in common with the SARS-CoV-1 virus is the ability to inhibit our interferon response. And so the idea is that if the virus can manipulate our immune response, inhibiting those interferons to get a foot in the door and then start to replicate, by the time the immune system catches up, it then launches this exaggerated innate response, all these different cytokines which then cause tissue damage in the lungs and in other tissues of the body. So this is potentially what's causing the ARDS. ARDS is acute respiratory distress syndrome. And the classic sign of this is a kind of over-inflammatory response of the immune system. And so cytokines are a necessary part of our response. They are small signaling molecules produced by the immune system, and not only, they're produced by lots of different types of cells. And their job is to help the body to adapt to a stressor, be it microbial or physical or emotional. They get released in these situations. And they trigger all sorts of physiological changes in the body. The problem is that when they become dysregulated or when they get released in an overly enthusiastic way, they can also start to damage tissues. And this is potentially what we're seeing here as well. The thing is that many doctors have been coming forwards and saying that the ARDS that they see in this SARS-CoV-2 situation may be quite different to the classic ARDS symptoms. What they're noticing is that potentially this looks a bit more like altitude sickness. There's more a problem of hypoxia than seen in other ARDS situations. And so it may not be to do with the actual mechanical capacity of the lungs to inhale and exhale, but it may actually be due with perhaps some clotting situation or blood disorder that is stopping oxygen from being able to travel around the body. So another thing to add here is that it's kind of like a double whammy with the ACE2 receptor, because the ACE2 receptor is also highly protective against lung dysfunction, against lung edema, against heart damage. And so when the virus binds to this receptor, it also takes it out. So not only do we get viral binding and entry into the cells, but we also lose some of the protective functions of this ACE2. And so then there are the viroporins. These are viral proteins like ORF3. These were shown in SARS-CoV-1 to be able to stimulate the NLRP3 inflammasome. The NLRP3 inflammasome is a collection of protein complexes inside a cell, and when it sees a threat, it releases a whole bunch of pro-inflammatory cytokines, as we've talked about before. The problem is that if it's overly activated and it produces too many of these cytokines, then we see this same overly inflammatory cytokine storm type of situation building up. And so they found that these viroporins in SARS-CoV-2 have a lot in common with the original SARS-CoV-1 virus. And the main cytokines that may be dysregulated here would be interleukin-1 beta and interleukin-18. So potentially raised interleukin-1 beta, raised interleukin-18 from NLRP3 activation, and raised IL-6 from increased macrophage activity. So then there are a series of proteases, the 3CL, PL, and M protease, as well as RDRP. These are a series of enzymes that help the virus replicate and evade the immune system. And they were singled out as kind of targets for drugs or natural substances in the original SARS-CoV-1. And there's also a lot of research going into how perhaps they can be inhibited in this situation as well. So potentially one of the main differences between that original SARS virus and this new virus is the addition of a furin cleavage site. So we have an enzyme in our cells called furin, which is used to activate proteins, and potentially this new virus is able to hijack that system so that it can cleave off the receptor and get a greater binding affinity to the cell. So this may be one of the reasons why this seems to be more infectious than the original SARS virus. So what about ACE2? Well, ACE2 represents a bit of a conundrum because on the one hand, it's a negative regulator of the RAS system. That means it plays an important role in lowering our blood pressure, and it does that by converting angiotensin II to angiotensin 1-7. Not only does it do that, but it's also been shown to be protective against lung damage, against edema, and against heart damage. So there's the idea that greater ACE2 expression might be protective in this particular situation, or in lung disease in general. The problem is that it's also the viral entry point. It's where the virus binds to get into the cell. So the opposing idea is that a higher expression of ACE2 may give the virus more windows to get into the cell and may actually make things worse. So just to say that researchers are still actively looking at the implications of either greater or lesser ACE2 expression. It's still very much up in the air. And there may well be other factors that come out which increase or decrease viral docking independent of the amount of ACE2 expression that there is. So perhaps the levels of angiotensin II, perhaps something to do with the AT2 receptor. And I put this out there because it's important to recognize that while we may be talking about strategies that could increase or decrease ACE2 expression, I still believe this is kind of a bit of a reductionist way to look at it, and we need to know a lot more about the general viral docking and the general situation and all these other factors before we can be definite about things to try there. So obviously the question is it possible to preserve ACE2 function while limiting viral docking? So let's talk about the risk factors then. So I'm sure we've all seen that with increasing age, there seems to be a greater probability of developing COVID-19. The point I want to make here is it really the chronological age, or is it the things which go along with aging? So we know that as we age, we are more likely to develop some of these comorbid conditions, hypertension, diabetes, cardiovascular disease, etc. There's probably also a greater chance of metabolic dysfunction, of low-grade inflammation, of immunosenescence, and quite possibly lower levels of vitamin D and melatonin and factors like that. So what I'll be trying to do is perhaps separate out some of the things which are chronological in nature, things that we can't change, and some of the things that are biological in nature, some of the things which we can change. So it may be the case that an 80-year-old who's fit and healthy with no existing diseases is going to be at much lower risk than perhaps a 60-year-old with a bunch of pre-existing diseases. So then there tends to be a slant towards male versus female. Amount of social contact is another important one, especially for caregivers and medical staff on the front line. Then being a smoker. So being a smoker, is it risky or is it protective? There seems to be some controversy here. And Dr. Chris Masterjohn did a great video about this, summing up this dichotomy. And the conclusion that he arrived at was that if you're a smoker, you may have a lower chance of actually being infected, perhaps down to the oxidative stress of the smoke, or maybe the effect of nicotine. But if you are a smoker and you do contract the virus, then you have a much higher chance of it going on to develop into something serious. So then there's the air pollution issue. Wuhan, the kind of epicenter of the initial outbreak, seems to have really high air pollution levels, as does the north of Italy. The north of Italy has the highest air pollution pretty much in the whole of Western Europe. And so I wouldn't be surprised if this turns out to be a big factor in this. So let's talk more about this idea of age. So as we age, our immune system ages too, and this phenomenon is called immunosenescence. So immunosenescence is a series of age-related changes that affect the immune system and, with time, lead to increased vulnerability to infectious diseases. So what are some of the hallmarks of this process? Well, some researchers have called this inflammaging, the mix of inflammation and aging. And generally we see raised levels of pro-inflammatory cytokines and CRP. There is reduced naive T cells, we have exhausted or senescent T cells, so T cells that are no longer doing their job properly. In some cases, they have seen an increased number of natural killer cells, but less activity and less cytotoxicity. Then there is an increased level of oxidative stress, which brings about a kind of remodeling of the immune system. And one factor that may drive this is that NLRP3 inflammasome. This may drive this phenomenon of immunosenescence. So what it looks like here is a general slowing down of the immune system, a general loss of function, a steady decline. But the point is that this is not necessarily inevitable. There are many different lifestyle factors that can modulate this process, that can both speed it up and slow it down. And in some cases, researchers have noticed an actual reversal in some of these parameters. And so I remember one study looking at exercise and the effect in elderly individuals, and what they saw was that those individuals who remained active had far less markers for immunosenescence compared to those who were sedentary. So those who were sedentary saw a picture rather like what we see on this slide, but those who were active seemed to have a much younger immune system. And this is true for all sorts of different lifestyle factors. So diet, sun exposure, getting good sleep, maintaining a generally anti-inflammatory lifestyle, dealing with psychological stress in a positive way. All of these factors can have a huge effect on the way that our immune system ages. And this was a study done in Japan. It shows Japanese women versus men, and they were measuring various different T cell parameters. And while we see a general decline in immune function as people are aging, and we also see a slightly less strong inclination in females compared to males, what I want you to notice from this diagram is that not all of the dots are along the line. There are many different outliers. There are some elderly people who have pretty much maintained their T cell functions, and there are some young people who don't seem to have such a good response. And so this should tell us that it's not just about chronological age, it's about the way we live, it's about our relationships, and it's about those underlying mechanisms like low-grade inflammation and metabolic health. So finally, let's take a look at inflammation, because if we want to understand how the immune system reacts to things, we need to understand how the inflammatory process works. Because we hear a lot in this period of like this exaggerated inflammatory response, the cytokine storm, the tissue damage, the sepsis-like symptoms, which is causing so many problems for people with severe COVID-19. But at the heart of it, inflammation is actually a healing response. In most cases is actually beneficial. It's just when it gets dysregulated that it starts to cause problems. So if I knock into a door or something, I get the classic inflammatory response. I get the heat, the pain, the redness, and the swelling, and that is part of the healing response, and that's mediated by these pro-inflammatory cytokines. So cytokines are small molecules. They are released by the immune system or also by other cells of the body, and their job is to create physiological changes in tissues to favor the healing response. So it might be vasodilation to bring more blood into the area, it might be increased permeability of capillaries, it might be fighting pathogens directly, and it often involves signaling to other parts of the immune system that they need to come in and take action. So then we have our anti-inflammatory cytokines. These are supposed to come and calm everything down and return things back to normal. And this dynamic response is essential. So we get a threat, we get inflammation, we neutralize that threat, and then we return to baseline. So this idea of threat doesn't necessarily just have to be physical trauma. It can also be microbial in origin, poor diet, poor lifestyle, also emotional in origin, psycho-emotional trauma, stress. All of these things can raise pro-inflammatory cytokines. So when I'll be going through talking about these lifestyle factors, one of the main ways in which they can be beneficial is that they help to regulate this inflammatory response. So we have low levels of these pro-inflammatory cytokines by default, and then we can have a strong measured response when we need it. And this is a hallmark of proper immune signaling. So when we're talking about about low-grade inflammation, when we're talking about metabolic syndrome, what we're talking about with the immune system is that it's losing the ability to signal. And it needs to signal because it's a highly complex network of cells working together towards an end goal. And if it can't signal properly, then it can't achieve that goal. And this is probably when we get a reduced response or a dysregulated response, which finally ends up in this pro-inflammatory tissue damaging response, which we see with things like the cytokine storm. It's a kind of last ditch effort to save the organism, and in many cases it may do the opposite.